structure of DNA. We have explained what is a nucleoside, nucleotide. Okay. Now let us inform the structure of DNA and RNA. So here is a ribose sugar. We have the OH here, OH here. Then there's a base over here. And CH2OH. CH2O P double bond O and OH. So this is one unit. We have to connect this with the next unit. Now the connection is between this oxygen, the OH group at 3 and the group at the oxygen here. So we will make the next molecule. Here is the connection with the next phosphoric acid molecule in the next unit. So we have double bond O, OH, O, CH2. Now this will be connected to the next unit from here. So we have the OH here again. And here comes the next base. Again, this is getting repeated over here. So this is the 5th carbon and this is the 3rd carbon. So the 3rd carbon of one unit connected with the 5th carbon of the next unit. So again here, PO, OH, OCH2. Connecting with the next. And here we have the next base. So these are the connections of the bases. And when we look at the structure of RNA, it is a single strain. A single linear strain will be there. But when we look at DNA, there is one more strand here on this side, which is bonded with this strand in a hydrogen bonded fashion. Okay, that means if we have a base which is cytosine here, then on the next strand This will be bonded with a guanine unit. And if we have a thymine here, this is, yeah, if we have a thymine here, this will be bonded with adenine on the other side. Now the bonding between the bases is hydrogen bond. And please remember, adenine and guanine are purine bases. Purine bases. And thymine and cytosine are pyrimidine bases. Meaning, purine base is bonded with a pyrimidine base. In the bonding, let us see how the bonding occurs. Here is a cytosine molecule. We'll draw the cytosine here. Nitrogen, nitrogen, a double bond over here, N, NH2 here, H and H. Then here there is a methyl group. Sorry, not a methyl group. This is bonded with the chain, with the ribose sugar. Here is a ribose sugar. Okay. Now here, these are the double bonds. Now, so this is cytosine. Now, cytosine will form a bond with guanine. So let me, on the next strand, we will have the guanine molecule. The guanine is keto oxygen, N, double bond, and here there is an NH2, and here there is N, 5-membered ring, and here there is H, and here it is bonded with a sugar molecule. I'm drawing the sugar in the reverse. I mean, the reverse way. Let me see one second. One second. Let me draw the sugar. Oh, here there is no H. One second. Let me remove this H. That's what I was getting confused. There should be a double bond here, N, and a double bond on this carbon. Yeah. Now, and you have an H here. You can see the cytosine and guanine. And this is connected with your next molecule of sugar over here. So this is one chain, and here is a second chain. Okay. One chain means, see here, this is further connected with the next step. See here, P, O, CH2, the sugar molecule. Again, O, P, O, CH2, next sugar molecule. You can repeat the same thing on this side also. Okay, CH2, O, P, like this you can repeat here. Let me make it a little. O, again P, O, CH2, CH2, here is CH2 and connected with the OPO, oxygen, OH, and then one second. This is the fourth carbon, and the third carbon, and attach it to the oxygen, one oxygen, okay. Again, you will have, no, wait, let me try. Just, so this, here I have the NH, and here I connected with this CH2O. Let me draw it. You should be able to draw this CH2O, P, double bond O, OH, O, no, this should be on the fourth. I should reverse it, sorry. Let me. So if I reverse this, Yes, yes, that's it. So OH here. Yeah. Now, now here is the next carbon. P double bond O OH. And here you have the next base. So here you have the base. This base and this base will interact. See that? Okay, just like here, one base is here, one base is here. They are interacting. Now, coming back to this side, cytosine and guanine. See here, then NH of the cytosine form a hydrogen bond with the oxygen of the guanine. The nitrogen of cytosine forms a hydrogen bond with the NH of guanine. And this oxygen forms a hydrogen bond here. So there are 3 hydrogen bonds in cytosine and guanine. 3 hydrogen bonds. In the case of, similarly you can draw the structure of adenine and thymine. And you can find that it contains 2 hydrogen bonds. Adenine and thymine contain 2 hydrogen bonds. That is because in adenine, the adenine structure is N, N, nitrogen here, nitrogen here. And then you have a double bond here. And what you have here, let me, if you check the structure of adenine, you have a CH3 yes. Yeah, you don't have any substituent on this side. And one NH2 on this side. There is one NH2 over here. H, H. And there is no substituent on this carbon. Okay. Now, this is adenine. And thymine, the structure of thymine, I draw here, this. How does the thymine look like? The thymine on the other side will be having this structure. We have oxygen, nitrogen. This is a pyrimidine structure. Nitrogen here, yes. And a double bond here and a CH2. Okay. And now, and this is bonded to the, here it is bonded to the nucleic acid, ribose sugar. Now you can see here, there is one site of hydrogen bonding, because hydrogen bond is between oxygen, nitrogen, no other element. And here there is an H. This forms a hydrogen bond with this nitrogen. And you can see there is no group here. This place there is no group. Only hydrogen is there, which cannot form a hydrogen bond here. And therefore, and look at the compare that with guanine. Guanine, we have one oxygen, one nitrogen, and two nitrogen. And the two nitrogen contain the hydrogen, and that forms hydrogen bond. In this case, you have only two hydrogen bonds. So the bonds between cytosine and guanine, CG bond, is stronger than AT bond. Adenine and thymine bonding is weaker because it contains only two hydrogen bonds, while CG bond is stronger since it contains three hydrogen bond sites. Okay, so this is the structure of DNA. And you should remember DNA is double helix structure. That is, it is something like a, a, what is that? A ladder type of thing. These are the hydrogen bonds. And these two strands are complementary to each other. Complementary. I have explained to you last time that how a messenger RNA is getting created from a DNA molecule. Similarly, here also in the DNA, if you have one strain containing different bases like A, T, G, C, T, A, like this, if you draw the bases of one DNA, very easily you can find what will be the basis on the other. So the complementary one with A is T, with T is A, G, C, and C, G, T, A, A, and T. Okay. Now two hydrogen bonds here, two hydrogen bonds here, three here, again three here, two here, and two here. So this is how the length of the chain increases. So this is the base pairing of DNA occurs. So we can say this base paired structure of DNA through hydrogen bond can be known as the secondary structure. Secondary structure of DNA molecule. Okay. And these strains will coil see one double helix structure. So this is one. And a second one here. And then here also the G of this will form a hydrogen bond with the C of the next, A with T of the next. Like this, two strains will bind together to constitute the whole molecule. So this is how a double helix structure of a DNA molecule is being formed. Now, what are the functions of biological functions of nucleic acids? Functions of nucleic acid. Okay. There are two biological functions which the nucleic acid perform. Number one, replication. This is done for genetic continuity. For genetic continuity. And this is the first thing that is occurring during the cell division. Okay. And also, so during the cell division, first the DNA will replicate and form an identical unit. And DNA contains all the genetic information. So DNA is the basis. It is the basis of, or we will say, chemical basis. It is a chemical basis of heredity. We say hereditary character, right? Heredity. And all the genetic information is stored in the DNA molecule. It is responsible for maintaining the identity of any organism. The DNA sequence in a human being will be different from that of animals and that of plants. So different species will have different DNA sequence. Okay. And this DNA, this is being transferred from the parent cell to the daughter cells without, in a very, very systematic fashion. And in this, the first thing is replication. So replication of DNA means forming another DNA strand. That is, a single DNA strand first will depair into two. So this is one DNA double helix structure. It will get depaired. and then a daughter complementary bases are formed. Complementary bases are formed on each of this, and new strands are generated on either side by accumulating material from the cell, from the cytoplasm. Okay, the adenine, guanine, everything will be synthesized inside the cell, and that will be put here, and new DNA strands are formed, daughter in the before the cell division. And once this separation and formation of the complementary chain is completed, then these two will form two nuclei inside the cell. And once they form two nuclei, the cell further get divides into two. So this is very briefly what replication means. So replication is a process in which the DNA molecule produce identical copies of themselves. So you can just note a definition. It is replication is a process in which DNA molecule produce identical copies, cut and paste, of themselves. So that each daughter cell, so that each daughter cell will have DNA exactly identical, will have DNA exactly identical to that of the parent cell. To that of parent cell. This is what is meant, what you can understand from what is meant by replication. In very simple terms, it get cut into two. Okay, two daughter strains. So this is the first function of DNA molecule. The second one is protein synthesis. Now, protein synthesis involves 2 to 3 steps. The first one, a DNA molecule will generate an RNA molecule. A DNA carrying the code of the amino of the protein. A DNA which carries a code of the protein. Code of the protein means protein, we have already seen what is meant by protein structure. That is ALA, GLY, what is the VAL, LEU. So this is the structure of protein, one by one after the other, it is to be arranged. And each of these amino acid will be known to the DNA as a triplet code. 3 bases will constitute one code for one amino acid. Next, three bases will constitute the code for another amino acid. Like this, the DNA carries the code. And this is translating this code to by constructing a messenger RNA. Okay, that means from this it will form a corresponding RNA molecule here. So if it is A here, okay, this is the A, adenine of the DNA strain. Correspondingly, it will construct a uracil over here. It will bring a uracil and keep it here. If it is T here, no, adenine, sorry, not uracil, guanine, AU. Then if it is thymine here, what is the corresponding thymine? One second. No, A, T, C, G, A, U, C, G. Oh, A will be having adenine, oh, thymine. One second. So if I have, let me make it more clear. Suppose I have a A. This is a DNA molecule. T, G, C,